Okay, good morning, good afternoon, or good evening, everyone, depending on where you're based at the moment. And a very warm welcome to our first CAT 3Rs training webinar in 2026. Today we will talk about NATworks, the new database I'm very excited about. My name is Kathrin Herrmann. I'm the director of the education program at the Center for Alternatives to Animal Testing at Johns Hopkins University, or abbreviated CAAT. And it's wonderful to see so many participants from different regions. And yeah, we are also excited to, for the first time, host a webinar in the time zone that is more accessible for Australians, people from New Zealand, Japan, and I think India is also pretty good. So in the US, it's 4 a.m., so that's not the best time. Um, so, and yeah, the webinar today will introduce NATworks, an industry guide to non-animal technologies. And it's designed to help researchers, developers, regulators, and innovators efficiently identify relevant companies and expertise in this rapidly evolving field. And the webinar will begin with an introduction to the NATworks platform. And then we have a series of companies that will introduce themselves. And then we will move on to a panel discussion with all speakers. And finally, we will have a moderated Q&A with you. And if you have specific questions for the representatives of the company, of each company, you can put those in the Q&A box and we will address them right after their presentation. But then later we will have bigger questions that are interesting for all of the company representatives. And yeah, this session is being recorded, as you could see. And also regarding certificates, that's a often asked question. We will send the certificates to everyone who attends the full webinar within about 2 weeks' time. And my colleague Anwen Stednik is going to do that from CAT. And with that, I'm very pleased to introduce our first speaker, who will introduce the NATworks platform itself. And yeah, Julia Ratzwell is a biologist and she's scientific officer at the German NGO Doctors Against Animal Experiments, DAAE, and she serves as the project coordinator for NATworks. And she has been closely involved in developing databases and tools dedicated to advancing animal-free, human-relevant research methods. Julia, over to you.
Yeah, thank you, Kathrin, and thanks for the invitation and yeah, for giving me the opportunity to present this newly launched platform. Yeah, it was launched nearly 2 months ago in November by our NGO. So, as you already said, it is called NATworks, and as you can see below the logo, it is an industry guide to non-animal technologies and what that means and how you can use it for your career building. I will explain to you in this short talk. Yeah, my name is Julia Ratzwell. I hold a diploma in biology. I'm working as a scientific officer for Doctors Against Animal Experiments. As you said before, I'm the project coordinator of NATworks. So first of all, we want to have a look at what this platform is. So it is a listing of companies that develop, provide, or apply human relevant methods. So either it's a product or it's a service or it is both. So it is freely accessible, so no paywall or whatever. And it has different target groups such as industry, professionals, regulatory agencies, researchers, and last but not least, and what we want to concentrate on today, individuals. So in regard to career opportunities. The platform is quite new. It is already registered as an EU transitional initiative. So that means it supports or can support the European roadmap towards phasing out animal testing for chemical safety assessment. Next question would be, why did we develop this platform? So as you can see on the slides, the various headlines, they all show a global trend, so all over the world. So some kind of shift away from traditional animal testing to human relevant and non-animal methods. For example, in the European Union, as I said before, there will be a roadmap published this year towards phasing out animal testing. In the US, the FDA announced a plan to phase out animal testing for certain drugs. The National Institutes of Health, they are going to prioritize human-based research methodologies. And as we have a guest from Australia here, Australia has got a national strategy as well on these technologies. So you can see there will be some kind of shift, and companies that apply and develop these methods will play a crucial role in this transition. Next question is, how can NATworks support this transition? So it can in multiple ways. First of all, raise awareness in general about animal-free science, human-relevant technologies, and educates about these technologies. A very important point is the access to non-animal products and services. So because there are some databases that cover these topics, but more the technologies and not list the developers and what is commercially available. And it can serve as a network and collaboration platform between people, institutes, and organizations, maybe for funding or for joint projects, for example. And last but not least, discover career paths. Maybe for people who may not want to stay in academia and looking for alternative place to work. Here we have an overview of potential users and the users. So for example, people who are working with animal experiments, they can find animal-free solutions if they're interested in switching or incorporating these methods. Again, for NAT developers, they can look for research partners. It's also useful for regulators looking for solutions and getting an insight on the landscape of the development status quo of the non-animal methods, and for early career researchers looking for employment. A short look behind the scenes. So how does this database actually work? And it's it is developed, so the company Freshie built the technical infrastructure, and with them we define queries. So handpicked queries like words or phrases that are important in this topic, and we're doing a web search with that to identify the companies that match our criteria to incorporate them into this database. And then different models are used to categorize these companies. And after that, AI is used to extract the respective data that is relevant for the database. So what we want to display in this database. And after manual validation, it is officially listed in this database. I've been talking a lot about what is it, why did we develop it, and now I have a short video for you to that you have a look at how this database actually looks like. It's a short one. I hope the audio quality is sufficient.
Around the world, innovation is already reducing the need for animal testing, but too often it remains fragmented and hard to find. NATworks changes that. Promoted by Doctors Against Animal Experiments and built by Freshie, NATworks maps companies delivering non-animal technologies in one open global platform, so policymakers, researchers, industry, and investors can see what exists, connect faster, and move the transition forward. Together, NATworks, making the ecosystem visible.
So you have an impression how this database looks like, and we want to have a look at three possible use cases that can be covered by this database. The first one is clients looking for a special product or service. So here you can see the interface of the database, and as you could see in the video, there are multiple filters you can apply. So when you're a client looking for a special product, you would choose one of these filters on the left-hand side. So you can search for products, you can search for services, and what is possible as well, searching for contract research organizations. Of course, you can search for all three depending on your tailor. Yeah, you can tailor your need with these filters. And another important filter would be the organ and system filter, because normally you are looking for a specific organ or, of course, research field. And by combining these filters, you get a list of companies that match all these criteria. For regulators, it can be quite useful. On the one hand, of course, looking for different methods, and for regulators, the most important filters would probably be the certifications filter. So you can search for ISO certification, GXP means can be GMP, GLP, depending, and you can search for approved test methods as well. And what else is highly relevant are the endpoints filter. So you can even search by endpoints and identify different companies that address these endpoints with their products or services. And moving apart from that, it can be very useful to get an insight of the landscape of technologies, for example, for identifying technologies that are still missing or are insufficient. And there can be special research projects be funded to fill these gaps and to enhance this transition to human relevant research. Yeah. And now how you can use NATworks for your career building. I guess most of you have a specific country in mind when you want to have when you're looking for a new job. So you can use the location filter. You can choose one country, you can choose multiple countries depending on your flexibility. And the second one would be organ and systems filter. When you worked with kidney organoids, when you're doing your PhD, you maybe want to stay in this area, and you can use, of course, whatever filters matters most to you, like research field, company size, or human-only companies. So for future developments, we want to expand and refine the search terms for animal-free providers. For example, this, we're thinking about some kind of label for companies that do not use things like FCS or certain antibodies. Education will be another important topic for job postings, internships, PhD positions, whatever could be done with a filter, or it's not defined yet. And we're planning a webinar series on careers in the sector. So basically, this talk is the first step to that. If you want to have a look at the database, you can scan the QR code, have a look, play around with the filters a bit, and that's it. So again, thank you very much for your attention. Thanks to Kathrin, and it would be great if you want to stay connected. Please follow us on LinkedIn to get updates and new events. If you have a specific question, you can send me an email. Please feel free to send that. And looking forward to the company presentation, and I hand over to Kathrin.
Thank you, Julia. That was a great overview, and thanks for actually making this database. I know how much work that is. And so we start with the company spotlights. Dr. Bettina Lickes is going to start. She's our next speaker. She's the co-founder and business development and scientific communications manager at InnoVitro. And she holds a PhD in neurobiology and brings over a decade of experience working with human iPSC-derived cell models supporting preclinical drug development through tailored human-based cardiac safety and efficacy assays. Bettina, please go ahead.
Yeah, thank you very much, Kat, for the kind introduction. Well, since it was quite nice and filled up everything, I won't talk too much about myself, but about our company and how you can find us on the NATworks database. And as you already have heard, we are a human cell-based assays provider, so a CRO for preclinical cardiac risk assessment. So we're quite in a niche. So if you look at the NATworks database, I'm just screenshotting here, I'm having no video. You have these different research filters, and when it comes to us, if you would like to, like Julia already said, you could look for the tissue, you could look for the research field. For us, you would always get a hit if you would put in the heart, of course, but also myocardium, if you would put into the research field the drug development, because we're working for the preclinical sector. Our country, we're based in Germany. That would also give you a hit, but we're actually working very international, so I don't think that really matters for our work. And endpoints, oh, I forgot to put that in, but something like cardiotoxicity, etc., would also give you the hit. And if you then go on our in vitro slide and enlarge the field on the NATworks section, you get a little bit more detail about us. You get, where we're based, where we're founded, etc., what kind of research fields we're in. Like I said already, the disease indications from cardiac diseases, atrial fibrillation, cardiomyopathy, everything is included. And also the assay formats, like cell lines, we're working with cell lines, we're working absolutely in vitro and using iPSC-derived cardiomyocytes in the heart model. So if you just want to really get in contact, you just click on our website basically, or you just use this in vitro contact page QR code, and then you land directly on our contact page and type in every kind of information from service relevance up to, like you also said, if you job opportunities, etc. You can type everything in. So just get in contact. So, but to give you a little bit more about what we do in depth is we like to cover the cardiac contraction cycle. So basically everything that happens within the cell with three different topics, or also three different devices. The first is the electrophysiology part, basically the way the cardiac contraction cycle starts. We use the EFP duration with the cardio excite system, where we are working very closely with another German company that is providing these Nanion technologies, and we're basically their CRO hand, and using the EFP duration for like an MEA system basically, and also for going into disease modeling like atrial fibrillation, which I'll hop into later a little bit. Our other field is the contractility. This is basically how we started. We feel very strongly about it. We have also, again, a 96 Watt system called the Flexite that we co-developed, and it has membranes instead of plastic surfaces, going into, like, being very physiological when it comes to preclinical screening. And also we then look at structural changes, because lots of compounds, etc., have not only an effect on the functional parts, but also have an effect on the morphology. And we have to kind of copy that as well. And we use another system for it. All of these systems that I talked to are real-time monitoring. So there's nothing going in, or no dye or anything is applied. So it's completely non-invasive. And we are on at least 96 Watt systems, or we even go higher than that, and we're quite physiological. So when it comes to the cell cultures, we work mainly with iPSC-derived cardiomyocytes, human-based iPSC-derived cardiomyocytes, and it actually strongly depends on the project if we recommend going for 2D cell culture, 3D cell culture, or even going up to organoid style. Everything is possible. And one topic why would we decide, okay, it would be more relevant to go organized style, is to think of the translatability of the data, although that's always a topic, of course. But the throughput is, of course, also very something you have to focus on, because that affects the costs. And costs, nobody likes to talk about it, but it's a very, very big issue. So everything combined, we either go for 2D cell culture or for the organoid style. And the services in depth means basically for the preclinical field, we provide a lot of safety pharmacology screenings, or a lot of toxicology testing and efficacy even. We also even go earlier in the drug development for drug discovery parts. And also we do proper cell line characterizations, because there are lots of people out there who don't use commercial cell lines, who do their own disease model, for example, or who do a normal cell line and want to know about the functional elements. Are they working the way we want to? So this is the cell line characterization part we do, and also we go into the disease modeling ourselves. So I thought, if I have enough time, I'm trying to be a bit quick, I pick one of each topic and show you a little bit what we do here. The first is basically a toxicity screening that we have done, focusing on the parameter of contractility, and we do that with the Flexite, as I said. And here you can see in the video the Flexite plate, which has membranes instead of stiff plastic surfaces. As I mentioned before, the cardiomyocytes are beating. That's why you see the membranes are lifting up and downwards. It's basically the view from below. And the electrodes, as you can see in the lower part of the video, are basically part of the device. So they basically measure via capacitive distance sensing how the contraction changes. When we look at contraction force early after contractions, beat rate irregularities, of course, but also the duration, the contraction slopes, and the area under curve. So everything is covered and being taken apart. And the first thing I'd like to use, I basically went straight into toxicity, into the chronic analysis, because it shows you very nicely how a compound can have an effect, but it can have a dose-dependent effect. It can have a dose and time-dependent effect. And with a chronic analysis like this, you can see that very nicely. So on what you see here in this level is basically different concentrations of erlotinib, which is quite cardiac safe, a tyrosine kinase inhibitor, and you can see the contraction force over 5 days. And you can see that actually only high concentration, a little bit is happening, but it's quite cardiac safe. But if you look at vandetanib, you see that we have a strong dose-dependent effect because it happens immediately, but then it stays pretty stable over time with vandetanib. And hopping into eribulin, you see that for the higher concentrations, apart from the highest one where you knock out the cells, that also happens, of course, but you see a progression profile over time showing that there are different kinetics that you can visualize in a non-invasive way on the contractility level. And we have not done that with 3 compounds. We've done that with plenty of compounds, as you can see here, with tyrosine kinase inhibitors, but also with anthracyclines, but also with tyrosine kinase inhibitors. And we have a little heat map here where you can see that we have quite stable effects highlighted in like greenish, but when it goes to toxic effects over like 1, 3, or 5 days, you can see that especially at higher concentrations with the 10 micromolar and longer time in culture, we do quite get a lot of toxic reactions with a lot of these compounds. Second little service that we provide a lot is the cell line characterization. When it comes to iPSC-derived cardiomyocyte cell line characterizations, people focus a lot on the CEPA compounds because these were initially used to get an overall view of how iPSC-derived cardiomyocytes perform, especially can they be used for pro-arrhythmic risk potential assessment. So the data that you can see here with high risk to intermediate risk and low risk using these compounds were then applied to the cells, and this is basically the data of the CEPA showing nicely how, for example, the fatylite is having a higher risk when it comes to pro-arrhythmic risk. And one of the cell lines that we were testing in-house were basically then put in the exact same frame and tested, and you can see also showing very nicely the risk potential of these compounds when they were applied to that in-house cell line. And last but not least, I hope I'm not too late already. And one of the disease models we were focusing on, with that we swapped for like the cardiac side that we also used for the cell line characterization to look at the extracellular field potential duration. And in this case, we actually also applied a stimulus to the cells because we can do that for each well. We have a reference electrode, but also the stimulation electrode and the recording electrode. And with the stimulation electrode, we provide a stimulus like a pacing for the cells for 24 hours for quite high, like 2.5 hertz. And this initial design was done by Seibert et al. And we basically copied it and put it onto the cardiac excite system. And after 24 hours of tachypacing the cells, what we see is then that, or what we wanted to expect is that the cells are basically adjusting to the situation by having an extracellular field potential, basically a duration shortening, which you would also see for the atrial fibrillation. And when we tested that for the data that you can see when we applied the tachypacing starting here, that we see within the first few hours, we already have an EFP duration shortening, which stays pretty stable over time, showing this a reorganization of the cells that we wanted to see. And when we're using something like dofetilide, which is like an EFP duration prolongation effect, basically trying to rescue this effect. That's what we basically applied from here. That's why we started with a 60% as well. And going up to the highest concentration, we actually match quite well the rescue effect and go back to 100% with the duration when we add dofetilide. So this was a little overview about what we do in general. I hope it was clear. And when it comes to job opportunities, because Julia said so nicely that this network base is not only about finding services, etc., it's also about connecting and finding job opportunities. Here's a little view of our core team. We actually just had two new girls starting with us, Britta and Hannah, and they are supporting our lab team, but we're always curious and we're always interested in interesting new people. So get into the contact box here and get in contact if you like. And I'm happy to hear about questions, etc. Thank you.
Thanks very much, Bettina. That was very interesting. And also, do you offer also internships or just short-term help?
Yeah, we have that on a regular basis, like students, etc. From bachelor, master, etc. Yeah, that's always part of it.
Okay, yes. That is great. Thank you so much. So I will continue with our next speaker, and we will have questions in the end because there were no specific questions at the moment. So our next speaker is Dr. Ilka Marschmeyer, and she is the Vice President of Business Development at Tissues. And Ilka has more than 15 years of experience in multi-organ chip technology and has played a key role in advancing human-relevant interconnected organ models for drug development, toxicity testing, and ADMET studies. Ilka, thanks for being here, and over to you.
Thank you. Thanks for having me presenting the work on tissues today. So our focus is really on multi-organ chips. So Tissues has been spun off from the Technical University in Berlin. We were founded in 2010. So now 16 years ago already, we have 30 employees, 30 associated researchers. We have a very strong customer base in both industry and academia, and 9 patent families with more than 140 patents. And also are ISO 9001 2015 certified in our quality management system, which means we are qualified or audited every year. So what makes Tissues different to most of the other organ-on-a-chip companies? So first of all, our focus is really on being as physiological as possible, as human-like, we can say. So we connect multiple and possibly also autologous organ models. It always depends on the readout, on the experiments you want to do, if it needs to be autologous or not. And very important for us, it's the near to physiological circulation system. So just imagine your blood, it's circulating, and like if you have too much blood, it will dilute everything. So it would be too much water to dilute everything, all the growth factor cytokines. They are not immediately washed out, but they stay in the system. And this is what we also want to emulate in the system. And that's very important for a physiological readout. We have the largest portfolio on organ models and assays on the market. Our chips are designed to be very flexible. So it's compatible with different organ models, different cell culture types and inserts. So it means that we can culture organoids, we can culture hydrogels in the chip, but we also can culture barrier models and so on. So it's very flexible and always with the same chip design, but we are also able to align our chips or the chip design to customers' needs. We are committed to a very high quality, also shown in the ISO certification, because we really want to pave the way for future assay validation. And of course, we also have an automation in place for fully automated multi-organ chip execution. So the components to our system are the organ models. We have a lot of experience with different organ models. We do not normally supply the organ models. We usually just supply SOPs. What we do supply is the chips, either as a product or we also offer services in-house. The chips work with an on-chip pump, so we really have like a heart that is pumping the medium around, circulating it. And the pumping heart is this device. It's just outside of the incubator, pumping 8 circulations at the same time with an easy touch screen. We have a heat support to keep the cultures warm for a short time, also outside of the incubator. This is what a chip looks like. So it's about the size of a credit card. If you want, usually it's a bit difficult with it. Yeah, so this is the size of the chip. You can see it. So we have a glass slide on the bottom, then a PDMS layer, but we also offer PDMS-free chips. And here you can see the circulation of a chip 2. We just have enough space because all our chips have the same size, the same footprint. So for the chip 2, we had enough space to just put 2 replicates on one chip, so they're completely independent. This area here is like our heart that is circulating the medium. It's just a pump that is putting pressure and under pressure on a membrane and therefore circulating the medium without integrating or without an inflow of medium, just like your blood that is circulating in your body. And these areas here, you can imagine like being one well of a 96 well, or in other chip formats, also one well of a 24 well. And whatever you put in a 96 well or 24 well, you can also put in here. Like I said, it can be organoids, it can be spheroids, it can be hydrogels or barrier models like a gut, skin, or lung model. And then you can connect it with a second organ model, same here. So this is also the size of one well of a 96 well. And then with the circulating medium here, you really have growth factors, cytokines, metabolites that are produced by one organ model really reaching the second organ. It's suitable for iPS-derived cells, primary cells, 3D tissues, and cell lines. We have an on-chip pump enabling the pulsatil flow. Like I said, we have the plug-in option for insert-based barrier models, and it's compatible with live tissue imaging, so you can always look at the cells at any time. It's fully microscopable. This is what it looks like in a laboratory setup. You have a standard incubator. You have tubes. These are 2 millimeter size or thick, so that's why you can just close the door and you don't have to worry about it. And then the starter, what we call our pumping device, just stands next to it. It needs compressed air supply and power supply, but most of the labs already supply that. So the Humamic MPS platform is there to emulate human biology. So this is our chip 4, which is really ADME and PBPK compliant. We have an intestinal model for absorption, a distribution by our channels, then metabolism by the liver, excretion by a second circuit, which is seeded with proximal tubular cells and glomerular cells to really show some excretion. And we can really show that some of the, like we tested it with several compounds, and we can see absorption. We can see distribution in our channels. We can see metabolism by the liver. And also, depending on the compound, either no excretion, if this is also seen by patients, or excretion into the urine circuit. So it really depends on the compound, and we really can emulate this ADME profile. So our chips are meant to be up to 5 connected organ models. We can also culture single organs if needed. Each is composed of multiple cell types. We have a physiological fluid to tissue ratio. We usually try to scale each organ to 1 to 100,000, but in some experiments we also go to 1 to 50,000. We have predictive modeling for translation insights from chip to human, and we have over 70 publications demonstrating the valuable results in collaboration with many of the leading pharmaceutical companies and also academic labs. So why is the multi-organ approach so important? So the multi-organ crosstalk shows to be required for disease models. We had a very interesting experiments with AstraZeneca in Sweden, where we connected liver and islets in direction, and we could really see, depending on if they are in single culture or in co-culture, the insulin production improved physiologically, and the liver glucose concentration also changed if they were in single culture and in co-culture. So it's a very simple combination of two spheroid cultures, and we really see a huge effect just by connecting the 2 organs, because they crosstalk and they connect to each other. Why is it important to be human? Because we've done some experiments where we had humans on a chip. Bayer here also in Berlin had a rat on a chip. just to show how the thyroid and liver co-culture differs in human and in rats. And it's very important to see, and it's a lot of people actually ask also for this comparison, even though we actually try to be as human as possible, because that's what we want to do. We want to compare our data to human and not to rat. But in some cases, we were not able to not do it. So in this case, actually, Bayer did the experiment with the rat. We did the experiment with the human thyroid and liver co-culture, and we could see significant differences, just as it is also in the living bodies. Why is it so important to have also the complexity of different organ models? Because the model complexity is required for physiological metabolism. So like the experiment with the 4-organ chip that I meant, we have proven ATME studies. So we have absorption, we have distribution, metabolism, and excretion on the human chip 4. Where metabolization of compounds also change the distribution, permeabilization, and toxicity. So just imagine a sub-compound needs to be absorbed first. Maybe not everything is absorbed. Then it's getting metabolized. Some of the metabolites are only produced for a short time period, and then they are also further metabolized. So if you just pipette and conditioned medium to another organ models, this metabolites might already have lost their reactive composition, right? So it's really important to keep these in the loop, to keep them directly connected. So next, I would like to introduce our Umimic Auto Lab. So we are here having a fully automated chips. This is inside of a sterile internal class 2 cabinet. We have a high precision liquid handling system included into their chip operation, and supply of consumables is supported here. A bright field and fluorescence microscope, so you can take images anytime. This is important actually for AI presentation or AI analytics, because you can produce a lot of data. That's usually a problem. If we talk to our AI experts, they always say like, oh, we need more data. And this is how we can support them with it. We have objective turrets of 1.2, 4, and 10 times. And the fluorescence filter changer. And also on the bottom, we have a 4-degree cold storage for up to 66 wells, which means we can really keep the cultures independent for a long time. And then just there's an elevator taking the plates up, getting them up here, heating them to 37 degrees. So then they can be pipetted anytime, and you can emulate morning, lunch, and dinner application of substances, always with each food application, or 30 minutes afterwards. If that would be done by a scientist, they would probably do it for a week or two. And if they're really enthusiastic PhD students, maybe even longer. But at one point, they probably wouldn't be too happy to do it mornings, lunchtimes, and evenings, right? So this is all done by an automated lab. And then our solution is also not to have only the chips and the auto labs, but also have an AI-based digital twin, so that we can have our MPS system, giving it to the auto lab so that we can have replicates very easily, and then having it also digitally twinned by an MPS system, which we can then always challenge with a real chip 4, so that we can produce a lot of data. We can challenge these data with our real experiments, which usually is not so easy with a real patient. And then once we have this really optimized, we can then translate it also into the precision therapy, into the patient, and have a lot of readouts. And just to give you an idea, so we can have the Umimic platform, get a lot of information sources like recorded data of CHIP4 health, time series data, directed measurements of SEFT sensors, clinical notes, and so on. Transfer these information, fuse them, predict them, do modeling, and then have a virtual Umimic platform, which we can then challenge with the Umimic CHIP4 and the patient again. And with this, I would like to thank you again for letting me speak. Happy for any kind of questions. And also, of course, always very happy to get some contacts and discuss more about organs on a chip. I can't hear you. I think you're still on mute.
Oh, sorry.
Okay,
Thanks
perfect.
for letting me know. Thank you, Ilka. That was very interesting, and there are many questions for you. But we are only taking 1 or 2 now, and then maybe come back to them later, or we
Sure.
can maybe look at them and type some answers. But one question is Tissues running programs on regulatory implementation of the organs on chips with the FDA?
Yes. So we are applying for several programs at the moment. Yes.
There are great opportunity currently.
Yes. Right.
Right?
Yes.
Yeah.
Yes. We're well aware of them. Yes.
And then there's another question. We take one more.
Mm-hmm.
Were any of the tissues chips already used for infection disease studies, such as viral or parasitic infections?
Viral has been done, but by another lab, and we don't have any data on that. So it's not our own data. I think there's going to be a publication soon on it, but like I said, it's not our lab, so it will only be disclosed to us once it's published. We have a tuberculosis chip where we have lymph node, lung, and liver autologous by the same donor. PBMCs from the same donor also circulating, where we actually try like vaccine the system first, and then after 14 days also challenge it with tuberculosis. So it's not a viral infection, but it's also vaccine.
So thanks a lot. Our next speaker is Professor Paul Adlard, Chief Scientific Officer from Tisara Therapeutics. And Paul is a neuroscientist with over 30 years of experience across academia and industry. And he's focusing on aging, neurodegeneration, and brain injury. And at Tisara, Paul leads the development and application of human cell-based 3D neural models to improve the predictivity and translatability of preclinical neuroscience research. Paul, please go ahead.
Thank you, Kat. That's a fantastic introduction. That's so good, in fact, I probably don't need to give the rest of this talk, so thank you. So Tisara Therapeutics, its platform is based around the real brain technology. So just to give you a very quick introduction as to what that is, effectively we have three core components. So we have two proprietary biomaterials, and then we have our cell population. So these three components come together in our benchtop robot to create this 3D hydrogel, which then over the course of 3 weeks of development in a standard incubator and using standard cell culture exchanges every few days, generates our 3D microtissues or mini brains. So these images here are just an example of where we've created mini brains using primary cells, using iPS-derived cells, and also using patient-derived cells. So once we've got our microtissues, then we can go and put those microtissues through our drug screening platform, where we have a variety of different things that we look at, from viability and cytotoxicity through to genomics and proteomics and other endpoints as well. So in terms of our technology, then it's highly versatile and scalable. So we can generate about 15,000 microtissues per day from a single benchtop robot. As mentioned, then our microtissues are ready in 3 to 4 weeks, and at that point we have mature functional neuronal networks. As you can see in this image here, then this is a 96 well plate, and so we generate either 96 or a 384 well plate formats of these microtissues, where there's one microtissue per well. And looking at the reproducibility and consistency between our plates, and we have a CV of less than about 5% for all the metrics that we look at. And one of the really cool things as well is that our microtissues are optically clear. So in terms of getting really high quality and really fantastic confocal imaging and things like that, then our platform is well suited to that. Our microtissues are also highly physiologically relevant, and you look at the genomic profile, it correlates strongly with the human brain RNA controls. And one of the good things about our microtissues is that the cells, once they are within the microtissue, they actually begin to degrade the original synthetic biomaterials, and they replace those with their own cell-secreted extracellular matrix. So we haven't done an extensive profile or characterization of that, but you can see down here that there's quite a lot of, the profile includes a number of the key components that you'd expect to see in a brain ECM. So we have, as I mentioned, proprietary biomaterials, and our cells come from just standard commercial suppliers. So in terms of what our microtissues actually look like, this is just an example over the first couple of weeks. So you can see on day 0 here, then it's effectively just tiny little dots, which are the cells sitting in the hydrogel. But you can see very quickly over the first week or two, then that cellular density and network complexity significantly increases. And as I said, we use these microtissues at 3 to 4 weeks of age. Most of the studies that our clients look at have been in this sort of more acute time frame, but indeed, our microtissues will sit quite happily in the incubator for 4 or 5 months, and we don't have any issues with diffusion of media or necrotic cores or things like that. So the platform is really good for both that acute and long-term type of study. So in terms of the actual microtissues themselves, we have about a 50-50 mix of neurons and astrocytes at this point. We don't have microglia in the mix at this stage, but that is something that we are planning on delivering this year. And all these various markers here are just an example of some of the different markers that obviously represent different cell populations and what have you that are present within our microtissues. And in terms of those cells, the density, the connections and synapses and so on, we're sitting at about half to a third of what you'd expect to see in the normal human brain. So in terms of the current models, we currently have 2 that are commercially available. So we have this arti brain or artificial brain. So this is our standard healthy brain model. We then use that as the baseline to create our AD brain for Alzheimer's disease. We also then have a host of other pipeline models as well. Perhaps I think the next model that will be commercially available will be our substance abuse model. So we have some really cool data looking at the effects of methamphetamine, for example, in our system, looking at both the effects on postsynaptic and presynaptic markers, looking at inflammation and other things like that within the brain, and show very good correlation with what you expect to see in the human setting. We also can develop and have done for clients as well, develop bespoke models. So clients can provide us bespoke cell lines or what have you, which we can then encapsulate into 3D microtissues. So I'll just very quickly run you through some services and client projects just to give you a bit of a sense of the sorts of things that we do. So we effectively do provide end-to-end services, from the very get-go of the project design through to the complex data analysis and then reporting that can be used for client reports, investor reports, IND filings, whatever the case may be. And we cover across a broad range of different services as shown here. And I'll just run through a couple of these as examples. So obviously safety and cytotoxicity is a big one. We use a lot of standard off-the-shelf kits looking at things for cell viability, cytotox, oxidative stress, and other outputs as well. And this is just an example here looking at an environmental toxin here with rotenone, looking at how, with increasing doses of that toxin, then you obviously see quite profound changes in the network complexity and cell density, which is shown here as well and recapitulated here with that viability assay. Looking at target validation and disease biomarkers. Again, here we are primarily focused on looking at our healthy brain model as compared to our Alzheimer's disease brain model. But again, we have a variety of different assays and readouts looking at things like basic imaging and confocal for various aspects to characterize those tissues, through to omics profiling with genomics and proteomics, secretome analysis, looking at FACS for secreted cytokines and things of that sort of thing as well. Now these data here are just data that we've taken from a client project, just to give you a sense of kind of what the data does actually look like. So we have a variety of microtissues here, which have then been treated with client compound. We have a bunch of controls here. And what this is showing you is the difference that we see in the A beta or beta amyloid burden. So this classical histopathological feature of the Alzheimer's disease brain. So you can see before treatment, you've got a lot of that protein mass here, that A beta in green. After treatment, there's a significant reduction, and obviously we can then go through and quantify that and look at how these compounds affect A beta burden or indeed any of our other endpoints. And of course, the cool thing about this, for this particular company, was it was a company not sort of in the neurodegenerative space. They were in another indication, but they were keen to see if there was any crossover with their compounds, and they came to us and were able to identify a new indication for them. So this is now looking at network and neural network analysis. Again, we can look at neural outgrowth and branching and various other histological-based outputs. And so this just shows you here the arti brain, or a healthy brain model on the top as compared to our Alzheimer's disease brain here on the bottom. So we can then use our own in-house algorithms to then quantify a whole variety of different features of those microtissues and of the encapsulated networks and so on. And this just shows you here, obviously, that as you would expect in the AD brain, we have a decrease in the numbers of neural networks and also a decrease in axonal length. So we've then been able to use that model. Again, this is for a different company, so a different client project, to be able to demonstrate that basically the client had a couple of different compounds. They were tested at a couple of different concentrations, and we were able to show them that their compound was able to increase the numbers of branches in the AD brain model. So again, in combination with a bunch of other data that we generated for them, it again supported their move into a new indication. That same company as well, this is just an example of some of the proteomics type assays. So we use the Olink HT Explorer platform that allows us to look at 5,500 different proteins in any of our samples. And this is just two, again, for this particular company, example of two specific biomarkers that we looked at. And I've chosen these because it really nicely shows how, in the case of the Alzheimer's disease brain here, as compared to a healthy control, there's a significant reduction in the AD brain model. And indeed, with that client compound, you can see a significant increase in that protein that brings it then back to the healthy control levels. And likewise here with this second biomarker here, then you've got a significant elevation in this case of this particular biomarker in the Alzheimer's brain as compared to healthy control. And the client's compound then brought that protein biomarker down again back to normal sort of healthy control levels. And those compounds, as you can see, have little no major effect in the healthy control samples. So again, looking at things, like NFL, looking at IGF, looking at GAT43, looking at all these other kind of classic kind of things that you might look at, then you can sort of look at those in these data. And again, these data were great for the company because it sort of uncovered a new pathway for their compounds of interest. This is just another client's work. This one was a little bit different. So, as I mentioned, we do sort of end-to-end services and do a lot of in-house work. We're also in the process of developing microtissue plates that will be shippable. So we'll be able to ship these out to clients. In this particular case, we've actually done work in-house for the client and generated the samples, which we then shipped back to the company so that they could do their own analysis. So again, we're very flexible in terms of how we're able to work with companies. And so for this particular company, they were looking at PROTACs to target tau, again, of relevance to Alzheimer's and other indications. And you can see here very, very briefly that effectively what we did was to treat with their compounds, then have a washout phase, collect samples, send them back to the company. And these are the data that they generated. And effectively, they were able to recapitulate a study that was already in the literature and also basically validate their new compounds that they were in fact able to inhibit phospho-tau as they had hoped. And then after that washout period, you can see there is this nice dose-dependent response where you get this return of the p-tau levels in those lower doses and so on. So again, really providing those sort of insights into efficacy and target engagement for the companies. Finally, this is again, one other company's data. So we do a lot of work looking at neurodegeneration and cell death, looking at various cell death pathways. One of the pathways that we've had a lot of focus on recently has been ferroptosis. So this is a fairly recently identified form of iron-dependent cell death that was originally identified in the cancer space, but which has subsequently been known to be involved in conditions like Alzheimer's, ALS, Parkinson's, Huntington's, head trauma, and various other indications as well. So it's really become of quite a keen interest in the neurodegeneration space. And so one of the cool things that we're able to demonstrate was that with our AD brain model, then that actually has an increased sensitivity, as you can see here, to ferroptosis, which you induce with this small molecule RSL3. So we have an increased LDH release, so an increased cytotoxicity in the AD brain as compared to the RT brain or the healthy brain. In both cases, then that toxicity can be prevented with the use of a classic tool compound here, this lipoxatin-1. But then we're able to then take that AD brain model and that ferroptosis assay and then again test a client's compound. So this is where a company has had a compound which has been used for a particular indication. We've then been able to screen that through this assay and actually identify that their compounds actually had efficacy in this assay. And the cool thing with that being that for the company, then it's a brand new indication, but also for the field, it was really exciting because this particular class of compound is one that's never been identified or used for the treatment of conditions where ferroptosis is relevant. So things like Alzheimer's and so on. So again, it's great for the company. It's great for the field as well. So that's the, just in summary, again, so this is our example of our microtissues. So we use neural stem and progenitor cells. Our cells are encapsulated in a proprietary ECM. They develop elaborate functional 3D neural networks after 3 to 4 weeks. We have all the presence of all the essential receptors for drug screening. Our tissue profiling matches that of the human cortex. They're assay ready within 3 weeks. 96 380 well format. Suitable for lots of high throughput type of assays and what have you. We've also done a lot of work with molecular devices, also a lot of work with AXAM as well, and we have a number of application notes out with them where the companies have leveraged our technology with their own in-house technology. It's all fully automated as well, so in terms of the robotic manufacture, but then also the media exchange and the maintenance of those microtissues, and very easy to integrate into existing workflows. And as mentioned, our Alzheimer's disease model is the one that's currently available in terms of disease model, but we will have other models coming online fairly soon. So with that, and this is just the team. Again, we're a very small company located here in Melbourne, Australia. We have a couple of our scientists. That's the scientific team here at the bottom, and obviously our management team, and we have a great advisory board as well. So with that, I will stop and pass it back to Kathrin.
Thank you very much, Paul. Great overview. There is one question for you about the lifetime of your 3D model. How
yes, in terms of the length that we can continue to grow them for?
Yes, and when you see apoptosis.
So yeah, our microtissues, as mentioned, so we they go in the incubator like any standard cell culture model that you just feed them every few days, and we typically use them at 3 to 4 weeks. But again, we've kept them for and we have looked at many months after that. So yeah, we can keep them in a longitudinal sense. I think the longest we've kept them in culture is I think 5 months or 6 months. And yeah, the only thing that really happens over that time is that they do start to contract a little bit. So, but other than that, they remain perfectly healthy.
That sounds amazing. Thank you so much. So we go into our next speaker now. And I would like to introduce Tobias Krebs. He's the founder and managing director of the company VitroCell Systems. And he's trained in industrial engineering and is holding an MBA. And he has over 25 years of experience developing advanced in vitro exposure systems for inhalation, eye, and skin research. Tobias, over to you.
Thanks again for the invitation to this webinar. Yeah, my name is Tobias Krebs. I'm the managing director of VitroCell. And VitroCell is a pioneer for in vitro exposure systems with the largest product program on a worldwide scale. We have over 25 years of experience in this special area. Our strong technical support team services customers in Europe. About 40% of our customers are in Europe, 40% in the US, and 20% in Asia. So we have a strong focus on research and development. So basically 5 to 8 new products are marketed, introduced annually. And our target industries are environmental research, the chemical industry, pharmaceutical industry, tobacco and vaping industry, regulatory authorities. And yeah, meanwhile, over 350 publications have been published by our customers. So what do we do? So our mission is to provide in vitro exposure solutions, inclusive the online dosimetry for all airborne substances. Yeah, all, you see here a picture of all breathable, inhalable substances that exist. And we guide them in a very controlled way to the target organs lung, eye, and skin. These are our 3 target organs, whereas our largest field, of course, is the lung. So here in the lung, we work sorry, it's a bit fast. We work at the air-liquid interface, where most traditionally the cells are cultivated in a submerged form. Yeah. So the cells are covered with cell culture media, which is very difficult for the substance to reach the cells in a controlled manner. And that's why our exposure technology has the air-liquid interface, where the cell culture media is provided through a membrane to the cell cultures from the lower compartment of the device, and the particles, the semi-volatile compounds and the gaseous compounds can reach the cells directly from the top, where the cells are not covered with media. So this is the most relevant exposure technology to date, because the aerosol maintains the original reactivity. And it enables also the integration of dosimetry tools. So here you can see the lung, and these are the cell culture systems, which are cultivated on membrane inserts normally. But we have now, together with our cooperation with the Swiss Biolix, the possibility to expose cultures also to a breathing lung-on-chip system, as well as the other organs like the skin and the eye, also in this chip system. So which application areas do we cover? So it spans of a wide range. You have the environmental health with indoor and outdoor pollutants. You have nanomaterial safety. You have bacteria and virus research. Chemicals and pesticides are tested with our products. Pharmaceutical evaluation takes place. Then you have the field of the tobacco and e-cigarette testing, and cosmetic testing. So if you see all those breathable compounds and substances, you have to consider different exposure principles, because not every substance can be exposed in an efficient way with the same technology. So here we have 3 groups. One is the continuous flow exposure. The other one is the single droplet exposure. And the third one is the dry powder exposure. And briefly, I'm going to show you some examples how those systems look like. So basically here in the continuous flow exposure system, you have a control module where you expose the cells to just clean air, paired with the exposure module, which then receives the aerosol plus a dosimetry position. So those systems, they reach from small installations like you see here to larger installations, which have then also a climatic chamber, up to 56 insert positions, as well as 96 well plates. So really we have a whole span of throughput, which can be realized with our exposure systems. When you want to sample from the environment at relatively low doses, you need longer exposure durations. And those exposure durations can be only achieved in automated stations, where you see here 2 examples, large stations which have a fully automated humidity control, which have a temperature control, and also the monitoring of all parameters during those 48 hours of exposure. So this ranges from large stations to also compact air tox monitors which have also a very easy to use HMI system. So basically this is used for emissions from industrial sources as well as from wood stoves here in this case. Here are some examples how you can use those stations. Yeah, so you can use them also for tobacco smoke evaluation, for environmental aerosols, for ship diesel emissions, for building material emissions, for nanoparticles. So all inhalable substances can be tested for over 48 hours in those stations. So what type of data goes online with those cell experiments? So first of all, I go a bit from the right to the left. The aerosol is analyzed chemically with a chemical characterization. The morphology of the particles is tested. Then also the physical aerosol characterization with particle measurement devices, and the actual dose on the cell cultures can be measured with built-in quartz crystal microbalances. And typical readouts are then a full omics profile of the cell cultures, as well as the basic assays such as cytotoxicity or proliferation or other relevant endpoints. So you have a full test battery then with all the dosimetry in an all-in-one system. The next product group is the single droplet sedimentation systems group, and that is the cloud systems. So the cloud systems you use when you have very small quantities of material available. Yeah, you need only 200 microliter of a substance to expose your cell cultures. And this is shown here in this short video. You have your cell culture inserts, you pip it in 200 microliter of liquid, you nebulize it, and in about 7 to 10 minutes, the aerosol is then deposited on the cell cultures. You can take the inserts out and then do your endpoints. Those application areas for this method are inhaled drugs, where you have very scarce materials normally. You have virus research, the toxicity testing of nanoparticles, and also the evaluation of chemicals. The third group is the dry powder sedimentation area. And this is when you have the task to assess smallest quantities of dry powder in the range of 1 to 100 milligram. So the range of applications is from inhaled drugs over nanoparticles to Sahara dust, which is collected, or as also in one of our customers' applications, moon dust collected originally from the moon is tested on salt cultures. And you can imagine here you have only really a very small quantity to assess the toxicity of this moon dust. So you have 4 positions here for inserts in the 12 well size or the 24 well size. And with high pressure, you then produce an aerosol from your small powder dose. So all 3 technologies can be also applied to the breathing lung on the chip. Here you see a continuous flow system, which has been validated already with gases and cigarette and e-cigarette smoke. Then you have the cloud system, which also has been validated on the lung on the chip system, as well as the dry powder solution. So with this package, we are a unique supplier of aerosol exposure technology to the lung on the chip and can cover all applications. This goes then in every exposure method online with dosimetry tools, where you have the microbalances, where you have temperature holders and other tools, which are a bit summarized here. Yeah, it's very important not only to just expose the cells, but to have also a clear information on the dose which reached the cell cultures, to have really then a full assessment of your experiment. With this said, I thank you for your attention, and I'm happy to take any questions.
So our final company speaker is Dr. Christian Maas, and he's a principal scientist at MPS Labs, a dedicated unit within ESQ Labs. And he's a physicist, and he's also a computational biologist, and he's specializing in digital twin platforms and PBPK-based modeling workflows that translate organ-on-chip and microphysiological system data into predictive human-relevant decision support. Christian, please go ahead.
Yeah, thanks again for having me, and I hope it's last and but not least, right? So we are switching gears a little bit. So far, great presentations from other companies, as well as Julia on the NATworks database, right? On well, how do you actually go about setting up these systems and producing data, right? But there is still a really long way from running a bioexperiment in the lab to making an impact into patients' lives. And that's exactly what we are about. We help to interpret, optimize, and translate MPS or any in vitro cell-based assay results, for that matter, to human and clinical relevant situations. Our focus is basically on patient kinetics and safety. And almost as a positive side effect of this is by using advanced systems such as MPS and organ-on-chip systems, we of course actually work very actively in the 3R field and have a means to actually save animals by either reducing animal experimentation or ideally completely replacing them. At the same time, however, and that might be more interesting to all of the bio and lab guys here in this call, is with our digital twin versions of your biosystems, basically, we can help to reduce sampling times, cost, and therefore, of course, the experimental effort. I'm a physicist myself. I have not spent too much time in the lab, but I hear stories of the pain the pain it is and takes to actually get data points by running these experiments. So anything that can make that a bit more efficient and cost saving, of course, is greatly appreciated. Our team right now comprises these 7 people, myself included, and we span ourselves a bit into like 3 main areas. It's of course strategy and business development. Then we have a data science, AI, machine learning team that's focusing on getting all of the big data together and then identifying clusters and patterns in this. And we just have the 2 girls here on the bottom side. They are Hannah and Anna, just joined very recently as well. And on the very right-hand side, Cleo joined as well end of last year. So the team is quite quickly growing now. We call this here PPK, or in general, digital twin, more mechanistic modeling. So really deriving and developing computational models that help you to analyze and understand why things the way they are. Within this, what we sort of like offer as an overarching tool is our virtual testing center. So this is the world's first software environment, basically, where you can analyze, interpret, and translate results from different pharmacological applications, kinetics, safety, and toxicity. But you can also translate those results to humans. You can classify your compounds or reanalyze your data and really get a good estimate whether what you see actually could be toxic in humans or not. But of course, I mentioned it, this is our micro approach here, where you can actually have simulations before you run any bioexperimentation that will tell you what would be ideal time points to measure and how often do you need to measure them. So this can reduce your experimental load and effort. With this, we actually could show that we can reduce the cost per experiment by about 60%. So this is quite interesting. We can improve the prediction of human relevant parameters, for example, liver clearance. So imagine you have a liver chip and you want to know what the clearance of a new compound is, right? And you translate that to humans. This is typically 5 to 10-fold under predicted. We can actually match that now very, very closely and improve that. So you know that what you see in vitro actually will be very, very similar to what you are likely to expect to see in humans. And we've done this with over 70 compounds on the kinetic side, across different studies and chip vendors. So it's not a cherry picking icing on the cake here that we are showing you, but it's really something that holds true across the board. What is a digital twin? It's a term that we used almost 8-9 years ago now already in this field, but with the recent hype of AI and machine learning, this becomes something that requires a bit more definition, I would say. And the way we think about digital twins is sort of like depicted here, and this is also the translational workflow that we typically apply for our PK and mechanistic models. So imagine you have an organ-on-chip experiment, and we saw some of these examples just in the recent presentations, and you apply a drug in this. So what we do basically is we turn physchem information, hardware information about how much media volume do you have, how many compartments, how many different cell types do you have, is there flow between the liver, and I think Ilka showed it right in this multi-organ-on-chip system. What's the distribution volume? How big is this volume with respect to the liver that's actually there, right? So all those type of information we take into account and put that into a mathematical description that allows you then to not only describe how a compound is distributed, metabolized, or stored inside the cells, but it also allows you to simulate how biomarkers are being produced and how potentially toxic effects have an impact on, CYP3A4 expression, right? And therefore reduce your clearance rate. So that's the type of information that we typically implement in this, and that can then be inputted into a human PPK or human digital twin model, and then, for example, you can predict, whether your compound should be toxic in humans or not. Just a quick overview as well. We also work in the chip performance assessment. This is something that we like to call as benchmarking. So here, this is actually the results from the liver ring trial that we run with tissues. It's a setting focusing on reproducibility of results. So this is quite a big topic. And we had here 7 or 8 companies that were supposed to follow exactly the same protocol in terms of setting up an experiment, preparing a certain drug concentration, dosing the system, taking measurements. So this was, followed by like a very unique protocol. And you still see, right, that depending on the compound and the company, there's still quite some variability. However, overarching with our analysis, we can actually show that the results are still, I think it's a 10 or 20% biological and technical variability. So this is really good to know, and this is something that we can, of course, analyze. So it's great that once you run those digital twin experiments, it's still the question of, okay, what does it mean for a human situation? So here in this call, we simulated and we wanted to predict how propranolol PK actually would look like in a human based off of in vitro experiments. So we first start off with developing this human digital twin model for propranolol, and what you can see here on the lower side in pinkish is basically the actual clinical data. So this comes from a real world example and publication, and we developed a computation model that can basically describe these data, right? So that's the blue purplish curve. Now, if you take the state-of-the-art approach, and this could be a simple in vitro 2D hepatocyte assay, or even a, company of your choice, super advanced liver chip experiment that looks at propranolol PK. What you get with a typical way of analyzing these data is a liver clearance that you scale up to humans that underpredicts the clearance, and hence you get a higher peak here, right? Because the compound stays longer than in the human system. And two things are really noteworthy here. One is that you totally over predict the actual Cmax. So this could mean that you would tag this compound as being more toxic than it actually is in the end, right? And that could hinder its effectiveness. On the other side, after 24 hours, you would simulate or you would predict that there's a bit of a residual compound here. So if you give propranolol now every day, you would start to see an accumulation, which in reality actually is not happening. So this is really it's a good example of a bad translation, basically. Now, with our approach, again, analyzing exactly the same data, we just have a more advanced mathematical model that can analyze this. We get something here around this black curve, which is not perfect, right? It's not matching the bluish, purplish dots, but it comes a lot closer to this. So this really shows you that how important it is to understand these numbers, and what you see in vitro needs a translation function to the human settings. How to find us if you're interested in this type of work on the NATworks database. I saw a comment that the slides are a bit too tiny. I hope this is better now. So if you just type into the NATworks main landing page in silico, you will find 3 companies right now. One is registered in Germany, and that's us. So that's ASQ Labs with its business unit MPS Labs. And we provide basically not only ADME-PK, but tox, different endpoints for almost any organ that you can think of. So again, if you're interested in working with us or finding us, then that's a really simple, easy way, next to, of course, visiting our website directly. Some of our partners to highlight what we are working on and how we work. So we have the majority of the bigger organ chip companies, as well as some bigger pharma companies. We have a lot of research grants and national as well as international projects that we are working on, but we also are very active in the regulatory engagement to foster not only education but also have every stakeholder at the table. Again, if you're interested in working with us, then I encourage you to visit our careers website. We always have open positions, always interested in internships. But it has to have a certain sort of computational background, of course, just as a disclaimer up front. And with that, I would like to thank you for your attention, and happy to take any questions.
Thanks a lot, Christian. Great opportunities at your company. That's very nice to see that you have so much capacity for new people, young people. There's one question one listener is asking, can you say a little bit more about the tissue chips assessment that you did?
Yeah, so in this little ring trial, I do believe there are 2 or 3 conference presentations on this as well, right? And there will be a publication at some point. So the idea was to, in the first setting, run the same compounds in a liver chip in tissues or with the tissue system to get the liver clearance value. The main focus was on how reproducible is this. So if lab A runs exactly that experiment and lab B runs exactly that experiment, how close is the number basically that you get out of this? And we looked at this, and within 10 to 20% uncertainty, this is basically the reproducibility across, I think, 7 or 8 companies and 4 different compounds. As an additional second step, we of course also looked at toxicity of 2 other compounds, but I believe that analysis is still in the making.
Okay, thank you so much. And now please may all the panelists switch on their cameras because we will start our panel discussion now. Thank you, first of all, to all our speakers for these excellent presentations. And I would like to start by asking each of you one question, and then we see what other questions are coming in. So, Julia, from your perspective as the coordinator of NATworks, where do you most often see the transition to non-animal methods breaking down? Is it awareness, access, regulatory uncertainty, or is it something else?
Yeah, well, I want to break it down to one single point. I think it's always, combination of different factors. So personally, I see major challenges in awareness and willingness. So when it comes to nanotechnologies, it is very often that it is emphasized that they cannot do this, they cannot do that. So there are many people, scientists, whatever, that are concentrating on what cannot yet being achieved with these methods and not what is already possible. Of course, not in, well, here we have companies that develop these methods, definitely not, but in general, so when talking about these methods, there is lots of skepticism. And I think it is very important that these methods become more prevalent in the general perception of people. So not only in the scientific sector, but also in politics and in the public in general. And this is why we have these databases. For example, we don't only have the NATworks database, we have the NAT database where these methods, specific methods are listed, yeah, to make clear what is, yeah, even now achievable with these methods. And I think regulatory aspects play a major role as well. So validation, quality assessment, and all this, yeah, I think these are the main factors when it comes to my opinion.
Yeah, thank you. Paul, do you want to add to this?
Yes, I was just going to say that I think there's a certain generational shift that needs to happen because, if you look at basic researchers or even, pharma companies and what have you, there's such a volume of historical data that most people have that's, relied on animal data. And, when you're progressing a particular story, you want to kind of, be able to compare your current data to historical data. So, I've worked with many pharma companies, and also having spent 25, 30 years as a basic researcher and academic, where you sit on these ethics committees and you listen to people justify why they can't use alternative methods, then it's a challenge. So I think we need the generations of scientists and forward-thinking companies to really sort of take up the baton and sort of try and make that shift.
Thank you. Tobias, what are your thoughts?
I think the validation of the methods is really, really key. We have seen today, again, a great set of technologies, of products which work already. But if they are not validated in a way that the regulators have a validated method in hand, which they can use for the assessment, somehow we have the risk that those methods get stuck in just internal company product evaluations and are then not used for the regulatory science. So I think our joint efforts must really be to really, with the stakeholders, discuss always to fund or to initiate the validation of the method. And, this is a quite cost-intensive and also time-consuming process. But I think it's very, very important to get those methods really accepted towards the regulators and to reduce in this way the animal testing.
Thank you. And Tobias, I just stay with you for the next question. From an industry and technology provider perspective, what tends to matter most to decision makers when they consider moving away from animal models? You already mentioned, of course, regulatory acceptance. What other, considerations are they putting forward?
Well, if you look from a company perspective, the interest is to have faster results, more predictive results, more this we can achieve with our human-based models already. Yeah, they want to have in a shorter time more predictive results at a lower cost. Yeah, to assess their product. Then, of course, on the other hand, the regulators must be sure that this data is really relevant. Yeah. And therefore, I think the regulators now started to challenge the system in saying, okay, we want the non-animal data, we want alternative method data, and triggered this a lot. So I think the initiatives at the FDA and also in the UK, they are really, really great, also at NIH. But this means, of course, that somehow, yeah, those methods need to have reliable data, and this is, I think, still a big effort for us. But yeah, this is what I think is very important. Yeah.
Okay, thank you very much, Bettina. In your experience working directly with clients, where do you see the strongest momentum right now for adopting human cell-based assays? And where do animal models still tend to persist by default?
That's a good question. So I think in general, what we see a lot at the moment is that there are, especially when it comes to the field of cardiac related questions, is that there are typical things that are still being addressed electrophysiology wise using classical models, not necessarily 100% animal models, but very classical models that's been around forever. And now they're getting complemented with other parameters, like, for example, the contractility, which wasn't a strong part, let's say, 5-10 years ago, but it has become more and more of the question because they want the overall view. They want to see everything happens in the cardiac contraction cycle. So that's coming more in now. And in general, I completely agree with what Tobias said before. And I think all these questions relate to each other. How do they implement and how do they moving forward at the moment? What I think is also important to see is from this point of view that we have this, let's say, this historical people, like Paul said, who are very focusing on the historical data, animal model wise. And then we have the new NAMs coming in. There's still some skepticism about it. What I think is always interesting is that people are easy to let's say, point at the things that we still have to improve about the human-based models, but we have the capacity to improve them, and we are improving them. Over the last 10 years, there's such tremendous great work been done to get better and better and better. But with the animal models, we still have there's the animal model, and there's the problems with the animal models, and there's literally nothing you can do about it. And I think this is now properly sinking in, and with the regulatory parts coming into it, there's the pressure now on all these companies to actually move forward with it because now the regulatory are opening doors.
Yeah, it's actually very exciting times. Paul, what did you want to add?
Yeah, just that I think there's a lot of obviously complexity around different organ systems as well. So yeah, my focus is the brain and neurodegeneration, and I think one of the big sticking points, for example, that we see is that a lot of folks are keen to see those kind of behavioral kind of endpoints that are perhaps more translate or immediately translatable, or they hope they are anyway, to those more clinical kind of outputs like learning and memory, or you might have sort of head twitch responses in methamphetamine abuse, for example, and things like that. So yeah, I think there's a lot of great sort of surrogates that we can use for some of those endpoints, but yeah, it's a big hurdle for a lot of folks to get over, is that lack of ability to do those classical behavioral studies that they're so used to doing.
And that have a very bad translational value, unfortunately. Yeah.
Exactly. I mean, as you're talking about before with the drugs and things, and the brain is particularly bad in the sense of that translation. And we know that across the board it's like a 90% failure rate. And if you look at things like Alzheimer's disease, it's even greater. At like 99% of drugs just fail in that clinical translation. So it's a real challenge.
So do you want to speak a little bit more from a neuroscience and disease modeling perspective? What do human 3D models enable that animal models consistently struggle to capture, particularly when it comes to translation? You already said a little bit about that, but can you expand for our listeners?
Well, I guess there's different aspects that at the end of the day, it's a human system. You're talking about they may not be perfect, but you're looking at those more human physiological responses. So again, mice, rats, those sorts of things are not human. Obviously, the classic example, certainly in my field, is Alzheimer's disease. That the vast majority of animal models that people use do not express the normal native human protein that's the pathological feature of that disease, and you end up with these models that are transgenic Frankensteins of sorts, where they overexpress multiple disease-related genes that just simply in combination aren't present in the human condition. So I think, at the end of the day, it's that translational and more physiological relevance. And also, and I think one of the other, I forget who it was, mentioned the cost as well, that in terms of the time frame to run these studies, and the cost savings and things like that are significant. When you are looking at these sorts of platforms, and that obviously has hopefully downstream consequences to cheaper medicines for people at the end of the day as well. So it's animal models have their place, but they have a lot of challenges in terms of how you can manipulate them and that ultimate translation.
Thank you. Christian.
Thanks. So a few comments, right? I would just throw them in. So I guess we as a community, we have we need to work on, there are two main groups, right? One is we simply have to show finally, and not only claim, right, that we are as predictive as animal models. We don't need to be any better. We just need to be as bad as the animal models, right? Because then you can directly replace them. That's step one, right? Step two is go do all of the basic research, right, and make these models better, right? Because we are still lacking understanding why compounds cross the BBB and why they don't, and how good is the model really that we have. So this is all really, really needed, right? I'm not against this, but I would see there are two distinct groups that we sort of have to work on. Cost is a factor that always is brought up, but I have the recent feeling it's more a bit of an excuse, right? So I mean, you can sit down and calculate the numbers. If you really find out that you can exclude two compounds that you would normally put into the clinical trials because you have a better predictive system a priori, right? You save a lot of cost, yes, but you still have to invest in the beginning and trust the system, right? And that's then on us again to convince people that, hey, we have the data already now, today, not in 5 years' time, to convince the FDA. They have or supervise make those experiments and want to compare animals, and that's the big part of the animal data in the bigger pharma companies. So there's no sort of easy way to get to that information, right? So I think sharing data while not sharing data with federated learning approaches, for example, that would go a long way, right? Because for us, it's difficult to find that one publication online where someone showed that an animal model did not work in predicting XYZ in a patient. So I think we either all work together, right, or we still try to fight that fight on multiple fronts.
Yeah, thank you. Ilka, when it comes to multi-organ chip systems, of course, they promise greater physiological relevance, but they are also more complex. Mm-hmm. What do you see as the key factor that determines if they are actually taken up in the drug development pipelines?
I think it's inevitable because we've seen it at the beginning. A lot of people said, oh, it's more complex. It's too complex for me to do it. I will start with a single organ or maybe go for 2 organ chips. And in the course of time, people actually notice, especially if we want to replace animal models, we cannot only look at one organ, but we need to see the different interaction between the organ models. We need to see if a liver is metabolizing something and these metabolites have an effect on my cardiomyocytes, on my intestine, on my skin model or whatever it is, right? So we really need to have this connection. I think there's a symbiosis of different methods, right? You always have to choose the method that works best for you. So you can start off with a single organ model, and then some of these single organ models, as we've seen today, are very sophisticated. So we can start with this. We can start also with a 2D culture first, and then move on and get more complex, more expensive. Also, in a 2D culture, I can do 348 experiments at the same time with a low budget and a low amount of work, right? But from there, you have to move into more complex buildings to see really what happens if the organs are interconnected. So choose your organ model or your method correctly. And if you want to go into the human, first you have to test it in a systemic context between the different organ models. And I see that there's no way around it, because we want to replace animal models. And the benefit of the animal models at the moment are that they are systemically connected, right? We don't see that in any other system. And that's where tissue is coming in to replace this lacking part so far.
Mm-hmm. Yeah, thank you. And so now we've heard several things already, but I want to ask you all, when you look at your different technologies and roles, where do you see the biggest remaining barriers to the broader uptake of NAMs and NATs? Is it, we heard about, of course, regulatory acceptability, then what about incentives? What about the research culture? Obviously, that's probably a big one. Where do you see the biggest remaining barriers in your fields?
The big goal, I think, for all of us is the keyword OECD method. This is really what the regulators like, and want to hear. So I think the process to get such a approved OECD method is quite time-consuming, also expensive, needs involvement of many stakeholders. But looking a bit at the funding scheme, I think more efforts should go into this direction to validate the methods which we already have. I think we have so many great technologies out there, and they work. We have shown this, but we must somehow get projects, support, funding. I don't know, from the EU here in Europe, and from companies and from other stakeholders to get those methods validated in a very professional way.
Christian, what is on your mind?
Yes, a lot. But I think I will keep it for this webinar short. So imagine you see someone walking on the streets with a super cool new iPhone, right? Think back, I don't know, 20 years ago when this was not a thing, right? And you see it, I don't know, in Australia, one person having this, and in Europe, one person having a new phone that you have not seen. So some people will be interested in, okay, yeah, let's see, this is a new tech, let's try it, right? But it took what, 10 years before everyone on the planet basically had either that phone or from another company, right? So right now, I think there's a handful of IND submissions with the FDA where in addition to animal experimentation, there is NPS organ chip or spheroid organoid data on there, right? And I think it was just recently opposed on the first IND submission with only 3 organoids in the onco area. So it's no wonder, right, that no one believes that our tech that we've been working on for like 10, 15, 20 years is the thing if we only have exactly 1 or 2 showcases. So something that I see like would break down the barrier is if we have showcases for like, 50, 100, 150 drugs where the same list of drugs is tested in all of the different organs that, you guys are all developing. And we have sort of like, a list for every organ, with every compound, and then you translate that to humans and you really see this drug would work, this could be toxic, the animal predicted this, the animals predicted that. That comparison is lacking. It's a bit of a grind, I know that, right? But like that's the easiest comparison in my mind we have to make in order to really prove to everyone and to ourselves that the tech we have is actually predictive. Right now, this is for the most still just a claim, right? That we can't back up. And that's a bit unfortunate because it's a really great tech. And I would really love to for this to take off. But it feels like we are postponing this moment until this actually is happening. And that's a bit unfortunate.
Okay, Ilka, what are your thoughts?
I would actually, I mean, I agree with both what Tobias and Christian said, of course, but I'd also like to point it out a little bit more positive, because I think we are really reaching a momentum at the moment, right? 15 years ago when we started, everyone was asking us like, oh yeah, but the FDA will never accept that, right? And then this is where we come from 15 years ago. And me personally, I didn't expect to see any changes in that within the next 20 years and maybe really have an FDA 2.0 already saying that organs on a chip can replace animal models. I expected to never see that in my life. I wanted to fight for it, but I didn't expect it. And I'm happy to see it, that this actually has been done in 2022, and that there are a lot of programs like the iStand and other projects now, even in the EU, pushing organs on a chip, pushing NAMs to regulatory acceptance, and also by the governmental issues to really say, okay, we are not accepting animal models any longer until 10 years. And afterwards, you have to have something else in hand. And this I can see is really putting pressure on people. And I see that it's also putting pressure on us to produce the best system to replace it. And now we also, we get into talk with other people, with pharma, not only those people who do it in science and do in vitro science already for 30 years, but we really get into contact with people who have been doing animal models for a long time. And we can figure out what do we need to improve the system to really move it to replace the animal models that you are using for I don't know how many years now. And this is really, really valuable. And we are really seeing progress going on there. So I also want to put it a bit positive, because I think we have a huge momentum and we will improve animal replacement a lot in the next couple of years. I really see that.
Yeah, it seems to be really unstoppable, but of course we all want more speed probably. And yeah, Bettina, what are your thoughts?
Yeah, I just wanted to hop on Ilka and Christian's thoughts, which are absolutely right. And there's momentum, like you said, and there's a lot going on. And I think actually this is also one of the things that sometimes I feel are a little bit overwhelming for some of them, actually, because then they say, when we speak to customers, we said like, okay, we have this issue, and actually we are thinking of, we don't really know how to address it yet with a non-approach methodology, and how. And then of course the question is, so how is your approach methodology validated, etc. Like the questions with OECD basically coming up, and yes, OECD takes forever, right? By the time it would be done, you would probably have a new technology in already. So a way would be, is of course going with the regulatory consortium, focusing on special things. That's what we do a lot. For example, we are taking part in the hazy chronic cardio tox study. We're part of the Botanical Safety Consortium, which is something very interesting for somebody who wrote a question about botanicals. Maybe that's something that is then of interest, because this is a consortium that focuses on using such methodologies for addressing botanicals in a safety way, right? Because that's actually there's no real guideline for it yet, and all these regulatory authorities and these combinations of companies, CROs like us, academics, and so on coming together, hopefully bring a little bit of a like structure as well, and helping people to decide, oh, this is my project, or this is the way to go forward for me, and this is the way to go forward for others. Especially, same with the NATworks platform, right? This will also help a lot to basically navigate in the jungle of new approach methodologies.
Yeah, thank you, Paul. Final thoughts for this question?
I think everyone's summed it up nicely, I think. And at the end of the day, I think it really comes down to time, and obviously getting enough time for people to appreciate what these models offer, and enough time to incorporate them into their own systems and companies, and to test them and things like that. And I think it's also important that we recognise that it's not going to be just a one size fits all. There's not going to be just one model or one system that predominates. There's going to be a whole handful of different models that are relevant and important for different situations, different indications, or what have you. So in the same way as there's 20 different types of electric vehicles or whatever, it's the same thing, right? That there will be a place for all these different platforms. And perhaps one of the other things as well is that if we are able to moving outside the sort of the basic science, moving outside the the pharma companies and things like that, if we're also able to get some of these technologies into sort of the government space as well. And I know we've had a lot of discussions here in Australia with the military and things like that, you know, there is a lot of interest in that sort of space in terms of using these sorts of NAM systems for you know, identifying, you know, sort of, or looking at medical countermeasures and things like that, and using it, you know, in the field to identify potential, you know, exposure sites or toxic, you know, exposure sites and things like that. So, and there's getting into that sort of space, again, I think would help really propel the uptake of those technologies quite a lot as well, I think.
Thank you so much. That was really wonderful. It was a very interesting discussion and yeah, inspirational.